Okay, I'm going to try and get this video done in one part. So we have one part for prokaryotes and one part for eukaryotes. We'll see. Okay, overall, this is sort of a big picture situation. You have ways in which eukaryotes are regulated because there's a lot going on here, right? You can regulate it here starting with the chromatin, right? And how the histones are unwinding. You have methylation, acetylation. You have things that you could do here. Then you could come here, transcription, post-transcription, right? The way we are processing the RNA, capping, tailing, introns, exons, the way it is leaving. Then you can translation level, right? How it's translated, the degradation of the mRNA. Then you can also regulate how your proteins are put together. So there's a lot of options here. Okay, let's talk first of all about epigenetics. This is the regulation when you are uncoiling your DNA. You did this. You did this with paper. We've thought about this. This is transcription and translation are physically separated. Regulation can occur at a bunch of levels, but the first level has to begin with what is called epigenetic, meaning it's related to your histones and your chromosomes, and it occurs before transcription even happens, right? Transcription factors can also be thought of as epigenetic. Just a little background, right? The organization of the human genome, you've got 20,000 genes. You have 23 chromosomes that carry those 20,000 genes. DNA is wound around histones. So those 20,000 genes are wound around histones. Expression of those genes must be unwound in order to transcribe and translate them. Okay? Yes? We remember this? Do you remember these pictures? Yes? How the chromatin is remodeled and remade and folded up controls transcription. So you know this. You need to be able to explain how this happens. You need to be able to describe how access to DNA is controlled by histone modifications. That's going to be your methylation and your acetylation. And you're going to describe how methylation is related to the epigenetic gene changes.
Function or activity.
All right, let's start that over.
6 refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
So that was just a fun little review of our acetylation and our methylation. and how it attaches to our histone tails. Okay, here's another picture of that. Right, here's your DNA wound around these histones, and it is called a nucleosome complex. Often the nucleosomes look like a bead on a string. See how they do? That's why it's called that, beads on a string, if you look at them on an actual micrograph. Okay, again, when nucleosomes are spaced closely together, right, when it's bound up like that, you can't transcribe anything. And that is when methylation of the DNA and the histones cause nucleosomes to pack tightly together. Now, when you have acetyl groups on there, histone acetylation results in loose packing, and it allows you, right, if you're looking at this gene, you can't get to it. If you're looking at it here, you can get to it. Chemical tags are added to histones in DNA. Sometimes it's a phosphate group, sometimes it's a methyl group, sometimes it's an acetyl group, and all of these serve as tags. They are not permanent. They can be added or removed. They act as signals to tell histones which regions should be expressed. Epigenetic regulation means around genetics, temporary changes to nuclear proteins in DNA that do not actually alter the nucleotide sequence. This isn't changing ATCG. It's not changing that. It's just seeing what we can expose to the actual transcription. Again, it's called epigenetics. It is the unwinding and opening of the DNA that allows those transcription factors to bind to the promoters and initiate transcription. Here's a lot of development in utero, environmental chemicals, drugs, pharmaceuticals, aging, diet, UV exposure. This can, if you look at it, their changes may be cancer, autoimmune disease, mental disorders, diabetes. These are all eukaryotic gene regulation at the epigenetic level. Okay, moving on. So we've unwound it. Okay, now let's look at regulation at the transcription level. Okay, so at the transcription level, you have enhancers, promoters, repressors. Okay, dramatic music. More dramatic music.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors.
Transcription factors are what we're talking about here.
RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase.
Any one of those things aren't there, transcription doesn't happen.
Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription
ATP
to
flying
begin.
in.
This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Let's just see how it's regulated.
Provides an overview of the major steps involved in RNA production.
In this animation, we will take a closer look at the events that prepare the DNA for transcription. mRNA and eventually proteins are produced when and where they are needed in an organism. Some genes, such as those for housekeeping proteins in processes like glycolysis, are expressed all of the time. These genes undergo what is known as constitutive transcription. Other proteins are only produced at specific times in certain cells. In these cells, genes undergo a process called regulated transcription. Transcription is regulated by proteins called transcription factors. These factors are proteins that are produced in the cytoplasm and eventually migrate into the nucleus, where they interact with DNA and activate transcription. These transcription factors only interact with specific genes, those genes whose transcription they control. Most eukaryotic species have over 1,000 transcription factors. Regulated transcription begins when a signal is received by the cell. This signal, often a protein, begins a signal transduction cascade that lets the cell know certain proteins are now needed.
Insulin, for
The
example.
most common activation event for a protein in a signal pathway is the addition of a phosphate group. That activated protein in turn interacts with another protein. This interaction leads to the phosphorylation of the next protein in the pathway. There is often a series of proteins that are activated by phosphorylation. At the end of this cascade, the final protein will be activated and then enter the nucleus. It must make this journey because transcription only occurs inside
In
the
the nucleus. You knew that. You knew that.
After entering through a nuclear pore, the protein interacts with the specific transcription factor responsible for activating mRNA production. As with the earlier proteins, the transcription factor is modified through phosphorylation. The activated transcription factor will next bind to an enhancer region. The enhancer is a region of DNA upstream of the transcription start site that binds a transcription factor. The transcription factor then binds to the DNA and moves to interact with the rest of the transcription protein complex, located at the transcription start site. When the formation of this complex is complete, transcription of the gene will begin. It is this sequence of events that ensures genes required for a specific tissue or only at a specific time are expressed appropriately.
Okay, transcription regulation. Gene transcription in eukaryotes requires RNA polymerase. Yes, we knew this. So it makes sense to regulate it through the polymerase. RNA polymerases require transcription factors, which again, we already knew, which are specialized proteins that initiate transcription. The factors bind to the promoter sequence, and that causes regulation. Eukaryotic RNA polymerases require transcription factors. So you have to have an RNA polymerase and transcription factors in order to get this party started. What else do you need? You need a promoter, right? A region of DNA, a few nucleotides long, and you need, come on, say it out loud, a TATA box. Yes, you knew it. Thymine and adenine, TATA. Yes, you knew that was going to happen. Okay, so what happens? Your transcription factor binds. Here's your transcription factors. Here's your enhancer that's going to kind of come around there, and all of this is going to activate. Where's your TATA box? It's in here somewhere. It's got to be in here somewhere because there's your RNA polymerase. Here's your RNA polymerase. Here's the first gene. Okay? Here's all of this that has to happen before your transcription can begin. RNA polymerase is in the proper orientation. Your promoter, everything is ready to go. You can see how this is going to be tightly regulated. You have enhancers, which is the DNA sequence that promotes transcription. You have, each enhancer is made up of a short sequence called a distal control element. And then you have activators that bind to that distal control element with mediator proteins and more transcription factors. There is just a lot going on here. We're not willy-nilly making mRNA that we don't need. A lot of things have to happen. You have to have transcription factors, mediator proteins, promoters, enhancers. All of that has to come together. RNA transcripts must be processed. Okay, so once we have all those things happening, so once this has happened and we're actually transcribing, okay, now what do we have to do? We have to deal with that pre-mRNA. It has to be processed. We know this. We just talked about this. Post-transcriptional modifications. We got to cap it. We got to tail it, and we've got to put some introns and extrons. We got to keep the introns in, so we got to cut them out so that the exons can exit with a cap and a tail. Of course this is going to be regulated. Highly regulated. Okay, so for this section, remember, you need to be able to explain RNA splicing. You need to be able to draw and explain 2 of the 5 types of alternative splicing that I'm going to talk about. And you need to describe the importance of RNA stability. And then here's some things that you need to define. All right, let's get started. RNA splicing. Okay, this is what we sort of told you in the last chapter. You've got these introns. We're going to take the introns out, and then 1, 2, and 3, and 4 are magically going to come together. That's not always the case. Sometimes we splice these things differently because they code for different proteins. Of course they do. So this is a whole level of regulation. You can have exon skipping. This is probably the most common form, especially in mammals, where exons are not included, right? You just skip over this. You just skip over this pink one, and only this actually exits. You can have an alternative 5' splice. So in this case, we've cut something else off. You can have an alternative 3' splice. So that is just which way it's going to happen. So here we cut out the pink one. Here, it looks like we went blue, orange, purple, cut out this altogether. You can have intron retention, which is a little bit crazy. An intron is not spliced out, and it's retained in the mature mRNA, which is a little crazy, but it happens. And you have mutually exclusive exons, a mechanism where only 1 or 2 of the adjacent exons is retained for the final transcript. So you can continue to modify this even after it's done. Here's another just example. So pick a couple of those that you like, watch some videos, and be able to describe those. How could this have happened? I would say splicing requires proper identification of introns. There could be arrows. This is a huge, if you want to think about how evolution could have happened, a lot of this, right? I mean, look at all of this that could happen. So one gene could do something else, could fold a protein a different way. It produces protein variants and doesn't really lose the original protein. So this could be involved in like hair color. The new variant might have an adaptive advantage. So you can see how this can link to evolution quite easily. Okay. The other thing we need to do is control of RNA stability. So what is that? That's our cap and our tail, right? Here's our little 5' cap. Here's our little 3' tail. And it is flanked by, they're called UTRs, which are just untranslated regions. So they're there for a reason. They don't get taken out. They're not introns. They're there for a reason. They're untranslated regions, but they have a purpose. That's why they're called UTRs. They can increase or decrease the length of mRNA, and they regulate mRNA localization, and it allows protein translation to start. So they know where the beginning is. Okay. The microRNAs, short RNA molecules, recognize a very specific sequence of mRNA. They associate that complex called the RNA-inducing silencing complex called RISC, and RISC binds and degrades the RNA. Okay, so you can watch this video these notes will be on there, but essentially it's just going through this. It's just saying these recognize a specific sequence of mRNA and they associate that complex to either induce or silence the mRNA. And in some cases, it can actually cause mRNA degradation. It can actually just chop up the mRNA. Okay. Initiation complex and translation rate. Translation is controlled by proteins that bind. which is the formation of the initiation complex, right? Our ribosome coming together, our 80 subunit and our 40 subunit coming together there. So how that happens, how that formation of the initiation complex happens, is also definitely going to be a regulation point. Eukaryotic initiation factors, also a huge point. GTP, which is our ATP option sometimes, which this complex binds the 40S subunit. Our GTP does that. Methionine initiator. What is methionine? The start codon. So methionine initiation tRNA, the beginning of translation, binds to the 40S complex. GTP is converted to GDP. Think ATP to ADP, and energy is released, and this starts the whole translation process. Okay, all of those things need to happen. So when it is phosphorylated, translation is blocked. When it is not phosphorylated, translation occurs. Right? Are you seeing phosphorus, methylation, acetyl groups? Are you seeing why we make you take chemistry? Right? A lot of things have to happen here, and a lot of it is based on chemistry, because it removes things, it changes the shape of things so something can continue to go through. Okay, chemical modifications. So even after we go through all of this and we get a protein, we've made it through, we've unwound our histones, we've got transcription factors, we've capped, tailed, and made our mRNA, it's left the nucleus, it's found a ribosome, the ribosome has come together, it's made a protein. Oh my gosh, we should be done. We're not done. No. Now, even the proteins themselves that we have so successfully made, even those can be modified. Chemicals can be added or removed. The way they fold can be changed. The way the chaperone proteins can be changed. The chemical changes regulate the protein activity or the length of time the cell exists. The changes can alter epigenetic accessibility. It goes back again and can regulate transcription or translation, all resulting in changes. Okay, I am not going to go over the cancer on the slides. We will talk about it a little bit in class. That is it.